Hello everybody. So I would like first of all to thank the organizers for the efforts to organize this meeting in these difficult times. Actually, I think it's my first talk in presence except of teaching for students since March 2020. So I would like to tell you a bit about our research on mathematical modeling of blood production system, which we now perform since 12 years together with clinicians, hematologists from Heidelberg Medical Clinic. And today I would like to focus on aspects related to evolution and function of intertumor heterogeneity in context of blood cancer, acute myeloid leukemia. So, but for this, of course, I have to start from the beginning, from the basic aspects of the modeling of the blood production system. So we are interested in understanding the evolution of the cancer, which is related to some mutations in the process of stem cell differentiation. So blood is one of the system which regenerates very well in adult tissue. So, and this is based on existence of adult stem cells. And in fact, it is blood for which the concept of stem cells was introduced for the first time. So this is probably also the tissue where the dynamics of stem cell is the best understood, although still not really fully understood. So the stem cells are those cells which can replicate and do self-renewal, and they are located in niches in bone marrow, and the process is you have this hierarchical structure. So this hierarchical structure, this discrete structure, we have there the different cell types which can be phenomenologically recognized. And the stem cells which are on the top of this tree, they can divide and they do self-renewal, but they also differentiate into different cell types. And finally, we get this sets of the functional cells which build our blood, immune system, and so on. And as I said, leukemia is a disease of the, in fact, disease of this blood production system, and it is due to some mutations in the production of white blood cells, and there the problem is that the cells replicate, so they get a signal that we need more cells, but they don't differentiate into functional cells, so there is accumulation of these blasts which are not functional. So to understand dynamics of this process, we focus on two of our cell lines, namely healthy granulopoiesis, so production of white blood cells, and this leukemic cell line. And somehow so it means we are interested in the dynamics of different cell subpopulations and communication among them. So if we look at this system, so we have to describe healthy system and we have to describe cancer cells. So we are here pretty lucky because we can look first at the healthy system independently. And this is what we also did, and this way also quantify it and test the simple models. Why? Because there are data on regeneration of healthy granulopoiesis from the patients who got, after some therapy like radiotherapy or chemotherapy, their white blood cells. So here you have data from different patients. And then their counts of this healthy white blood cells dropped. Actually, they decay immediately after this therapy, and they get a transplantation of bone marrow. So this is a mixture of cells from bone marrow, which is supposed to contain stem cells, but also early progenitors. It's also important for the time of the reconstitution. And then within 2-3 weeks, the patient recovered their blood. And of course, this is also interesting problem from the clinical perspective, because during this time their immune system is not working properly. So there are all these questions: how to make it faster, can we make it more efficient, and so on, so on. But here I will just focus only on the using of this data to test our model for the healthy cell line. So the model is relatively simple. Namely, if we consider dynamics of such cell compartments, we just describe their basic processes. So what they do, all cells except the last compartment, mature cells, they divide at the rate p. They produce 2 daughter cells, and with probability A, or we have our deterministic macroscopic model, so the fraction A of the daughter cells stays the same as the mother cell, as you see here, and 1 minus A cells differentiate to the next compartment. Yes, so then we have this differentiation, and for the last compartment we will have just only influx from the previous. We don't have any divisions, and we consider death. We could also consider death rates for all compartments, but as they are also biologically not really identified, and also mathematically it does not change the structure of the model at all. So interesting remark is, of course, when we work with these models, we don't at all recognize between symmetric and asymmetric divisions on this level of the individual cells. We have just these fractions which tell us which proportion of the daughter cells stay the same as the mother cell and which goes to the next compartment. And we need, of course, to describe, we have dynamics in which we see this convergence to homeostatic state, and then we definitely need for such models to consider feedbacks. Okay, so for feedbacks, of course, as in most cases in biology, it is not really well understood, but there are different cytokines, different regulators of the dynamics, and here in this model we consider just cytokine which is produced by other tissue and just used up somehow by the cells in the bloodstream. For simplification, we can consider just only last compartment for this degradation. And since this dynamics is much faster than the proliferation, we can use here quasi-steady state approximation. We can do it also rigorously using Tikhonov theorem, obtain this quasi-steady state approximation, which tells us how the signal intensity depends on the concentration of mature cells, which is this kind of Hill type regulation function, also often used in applications. And then the question was, is it self-renewal or proliferation of cells which adapts due to the lack of the mature cells? And of course, more intuitive would be that the cells divide faster if you need them. But model shows that, in fact, the most efficient is to regulate self-renewal. Regulation just of proliferation would be very inefficient. You would need 1 year to recover the blood. While regulation, so here you have comparison of these different regulations. The regulation of the self-renewal is a very efficient process. Of course, if we regulate both, it's even slightly better, but we can focus on a regulation of our A parameters. And what it means that if we need to recover the ultimate compartments, at the beginning, it makes sense first to replicate much more the early compartments, and this is due to the fact that we have very few cells in the early compartments comparing to the late compartments. We need to count from the 10 to the power 5 to the 10 to the power 9 in case of blood. That's why the self-renewal in blood must be very efficient. Okay, just as a matter of fact, for all fans of structured population models, of course, this kind of maturation could be also modeled by a continuous model, and we developed and analyzed such model with Benoit, Marie Doumic, and Georges Zubelli now more than 10 years ago. Here, for applications, we focus on the ODE models. First of all, because the differentiation seem to take place during cell divisions, and these models can be also in many contexts handled better, and at the end, I anyhow reduce the structure to 2 compartments. So what we could do with this model of healthy cell line, we could quantify it. First of all, in the upper panel, you see this data which I showed you before for individual patients. Now we divided them, because if you look at all data, you think, okay, for some patients it's monotone, for some you have overshoot, for some you have even damped oscillation. Why is it like this? And it's interesting that you can recover all these types of dynamics with this model with very small differences of the self-renewal parameters, which even cannot be measured in practice. So somehow it is this heterogeneity, interpatient heterogeneity can come really from the differences in the self-renewal of the cells. We could also, in the lower panel, we compare the model to clinical data for the large patients group, saying which percentage of the group recovered the blood, came to the equilibrium in the certain time region. So it seems that the model is not entirely bad. And okay, this is a relatively simple model. We have a system of ODEs, nonlinear ODEs, but just for the information, as it's important now for the different extensions of the model. In this model, we have just a unique positive equilibrium, which is independent now on the number of compartments, always, okay, no, which is asymptotically stable. And the trivial or semi-trivial, if they exist, they are unstable. If we have 3 or more compartments, we could have also a regime of Hopf bifurcation, so then we would get oscillations. But interestingly, this regime of Hopf bifurcation is far away from the parameters that we estimate from patients' data. And you need to change at least 2 parameters to come to this oscillatory dynamics. And we all know that oscillatory dynamics is also often relevant for blood diseases, but it seems not to be the case now for the problems which we consider here. And for, interestingly, for rigorous proof for global stability, we have only for 2 compartments with this pretty fancy Lyapunov function. I think I also said more than 10 years ago that I will give a prize for Lyapunov function for more than 2 compartments for this model, because we spent ages on this and could not solve this. Okay, so this was this model. Now, if you want to extend it to leukemia, the simplest way is just to describe again leukemic line with a system of ODEs. And of course, the simplest idea is how they are connected. They are connected through the same regulatory signals. So it means there is kind of competition for these cytokines. One can say also, or maybe cancer cells don't respond to the healthy cytokines. And then we can also neglect it and then have this kind of competition between different cell lines for the space of the niche. I will not continue on these 2 models and comparison. We did a lot. We even showed that it seems that both mechanisms work, and for certain individual patients we can even sometimes identify which mechanism, and it has also consequences for the disease dynamics. But we don't have time now for this. I would rather go to the heterogeneity. And before this, just to tell you again, the model is simple. And even since, as I told you, dynamics is similar for multiple compartments, we quantified model with realistic number of 6-8 compartments that could be cell types that could be recognized experimentally and so on. But you can also feed this model with 3 compartments and even with 2 compartments, because as I said, for this patient data, we are far from this hub bifurcation regime. So we work now with 2 compartments. What means we have our most important parameters are 2 parameters, self-renewal and proliferation. We can say of stem cells, we can say also of just dividing cells, but the largest self-renewal is the self-renewal of stem cells, so in fact this is the most important parameter. And then we take data of patients, so you have here data of patients. We need for this, I don't now go to details, but we need for this also data with relapses, so that we have also time between diagnosis and relapse. And we can fit, so we have, because this way we know the dynamics, we know how long it took to develop to certain level, and then we can feed the model to this data. For single patient, we don't get a unique parameter identification, what in the space of self-renewal versus proliferation of this dividing compartment, for each patient we find a curve. And you know, you don't get this uniquely because in this first equation you have just A times P. So somehow this is what you can estimate, not more, from this kind of data. So these are data for different patients. And then we compare this estimates with the disease dynamics. This was in case of patients who already passed away, and we knew what was their survival. And this is interesting that the patients for whose the estimated curve was more located in this upper right corner had much worse course of the disease. So they passed away significant. This is statistically significant different. So again, it seems that the stem cell self-renewal and proliferation parameters are essential for the dynamics, for the dynamics. And we have also data of patients with multiple relapses. And interestingly, for the next relapse, if we estimate parameters again, this curve shifts into bad direction. So this seems that the next relapses can have much worse properties, and this is a little bit links to the development of the resistance of the disease. Now we can move to the clonal selection and clonal evolution. So what are these clones? So clones are the cells which stem from the same mother cell. So it seems that now, especially if we have mutation, so then we have the whole clone which carries this mutation. And these investigations were motivated by 2 Nature papers in 2012, where for 2 different acute leukemias, AML and ALL, they observed, and they were surprised observing relatively small heterogeneity, clonal heterogeneity in this cancer on the level both of diagnosis and relapse. So like on the level of diagnosis, for example, they see only 3. Usually they saw between 2 and 6 different clones. And on the level of relapse, also few clones. And sometimes they were the same clones, but sometimes they were different clones. And one of the questions was, do you need mutations in between to understand this? What is the evolution? And for this, we extended our model to these multiple clones. And first, I will just speak about ODE and then switch to the continuous model. So in the simple situation, now our dynamics of such single line, as I said, we just reduce it to 2 compartments, but we may have now multiple compartments for different leukemic clones. And we just simply parameterize them with different A's and different P's. And first, in simulations, if we just want to mimic the evolution, we start with cells which have certain self-renewal and proliferation. And then what happens if we would put, okay, here in these pictures, you see simulations for 50 patients put together. So there are, that's why there will be always more cells than you see in single patients for the selection. But if you then would say, okay, then there are some mutations where we have appear single or small amounts of cells of very different properties, how the system evolves. So if we let such system evolves, then we observe selection. Again, these are here aggregated data for 50 patients. We observe selection of the parameters with relatively high self-renewal and proliferation. And then you can also simulate this relapses. So it means first there is chemotherapy. So this is cytotoxic chemotherapy. So it means we just add to the equation the term saying that the cells are killed proportionally to their proliferation rate. And then we simulate this till, okay, so I did not say it. So in fact, what I show here are the cells, the clones, which have a mass which could be recognized. So I don't say that, and of course there are no single cells of different types that are left here. But these cells, they really build the tumor mass or the cancer mass. So then we simulate this chemotherapy, and then as in the reality the cell counts drop. We don't see them anymore, but then we again allow system to evolve, and then we observe relapse, which is also having high self-renewal, but somehow the selected now clones have lower proliferation because, I mean, the guys with high proliferation were those which were killed, and in simulations, if the counts were just coming near zero, we just remove this clone. So we see the dynamics just in simulations which somehow resembles this what is observed in the data. So then, can we understand it better? For this, we just to be independent of the number of compartments, of the number of clones, we put the model in a continuous version. So now you have continuum of these clones. So it means we have the whole cell population, and we structure them by their clonal properties. So we have now an abstract variable x, and then this x determines a parameter and p parameter. So one could also just make it directly instead of saying that we structure everything by A, but this way we have just also, I mean, we can have just one-dimensional structure and both parameters structure dependent. And if you look now, okay, these are the simulation of single clones, there could happen that some clones, as we observe, grow earlier, and some clones, but then decay, even without chemotherapy, but some will emerge later. And if you look at equations, and in fact this is very well linked to this type of problems that Camila was talking on Monday, because even if you look at the first equation, it has a similar structure to these equations, just regulation. So these are systems with competition for this through this non-local term. So ρ2 is the total amount of V, and somehow you see from the first equation that ρ2 is the same for E independent on X. Yes, so somehow the value in the brackets just depends. The differences between different X's depend on A, and if ρ2 is small, then this bracket is positive, so you have this exponential growth. But then, if U is growing, V is growing, then ρ is growing. So somehow the bracket. So you come to zero of this bracket value, and then where you come there, of course, first for the smallest A. So for the smallest A, then you will get decay. And it is also already, I would say, intuitively that you can expect that for the highest A, you will have, this will be the only which stays and grows. And this indeed we can prove rigorously. So we can prove that the solutions converge to measures that are concentrated at the points corresponding to a maximal value of the self-renewal. So somehow this proliferation is completely not important here. This only says how fast is the process. Even in the analysis, you can scale it out, and even if it's x-dependent, and just forget about this. So this is a parameter which is important. Of course, if we have isolated maxima of the A function, so if here, if it was in one point, you have selection, very fast selection to such weighted Dirac. If you have the same in 2 points, you may have 2 of them. So what is here may be interesting that, however, to approach this analytically, we had to find another way to prove it, as in this previous works of Benoit's group and what was presented by Camillo, because here we deal with the system of equation. And we deal with the system when we cannot do really estimates. We cannot see boundedness of U just only by first equation. So we somehow, the dynamics is complex this way that at least I don't know if one could reduce this to the other method. But what we did, just very shortly, so you need boundedness and strict positivity of the masses. And here, I mean, this is a little bit tricky because, as I said, you cannot get it from single equations, but you can look at equation for the quotients, and then just with several tricks come to this proof, and so you get boundedness and strict positivity of masses, and then just looking at equation for such quotient, we get exponential decay of solutions to 0 in all points except of these points corresponding to maximal value of A. And from this we get this convergence to Dirac measures, and then we actually get convergence to stationary measures, also in a bit tricky way. Namely, we we show this convergence to the weights corresponding exactly to the ODE with maximal value of A. And how we do it, we just notice that, in fact, if you have a system of ODEs and you add perturbations to and this system have Lyapunov function and have global stability this way, but then you perturb it by some function which has good properties, is just L1 in R plus, so we have this decay to 0 of this integrals, then we have the same Lyapunov, we have convergence to the same equilibrium for this perturbed system. And in fact, in our case, it is working. So if you divide this way, you get the same dynamics as for this ODE system, and that's why this Lyapunov function is so important to have it. And about convergence, it is as in Camillo's talk, we have all this weak or weak convergence in measures, or narrow convergence, depending on which definitions you use for convergence. We can also speak about convergence with respect to specific metrics, for example, the bounded Lipschitz distance flat metric, but these are just technical details. So somehow we could recover with our model this kind of observations that there are only few clones. Of course, observations are infinite times, so you may still see more clones, and that some clones may disappear, some clones may appear because at the beginning they were very small numbers of such mutated cells. And we could also compare it a little bit to the patient's data. For patient's data, we don't have quantities of different mutations, but somehow for example, in one case we knew that one mutation was present, the other was not present at the beginning but appeared later, and qualitatively just as a proof of concept, we could recover it also with the model. So somehow for this, if we have the models which we could well quantified now, we could say for these mutations, which for clinicians are just they don't know what the cells are doing. They only have some molecular characterization of them. We could say, okay, this green guy will have the highest self-renewal, for example. Or we could estimate these parameters. Just for information, so I said we did everything for 2 compartments with the selection. We extended this result also to model with multiple compartments, having also these intermediate stages, because the question was, is this selection only depending on the stem cell properties? Can self-renewal, heterogeneity in self-renewal of progenitors influence this? And this was work with Tommaso and Thomas Stiel, and we showed that in fact nothing changes in this sense that we also, here you have time, and here you have these different clones, and here this is this with maximal A, and you have selection, or if you have the same A, you have selection in different points. And of course, if we have more than 2 compartments, we have to do it outside the Hopf bifurcation regime. Otherwise, we observe numerically that there is convergence to measure localized in this point, but of course then it's not a stationary measure, then we have oscillation as in ODEs. Okay, so this was for the selection. I did not consider mutations so far. So somehow we showed that if we have mutations at the beginning, we can describe this heterogeneous dynamics. But of course, mutations are there. And now I would like to tell you a bit what we did for mutations. We just went from 2 sides to understand the story better. Namely, first, what they can really measure. So what they do, they take the patient sample, and for the patient sample, they just know how many mutation different samples have. And of course, what we would like at least to get from this, to know from this what was the evolution. So what is the clonal hierarchy? So maybe more exact. Okay, so when we started doing it, there was still single cell analysis was still too expensive to be really done, especially in the clinics. But anyhow, these are rather experimental works than usual patient data. So here they have this bulk data. And in this bulk data, what they know what is frequency of different allele related to different mutations. And for example, in this sample, we have 84% of blast at diagnosis. So 84% of the cells are cancer cells. And we observe, for example, for this purple cells, that this is in 42% of the here identified in 42%. Since we have heterozygous mutations, we can from this conclude that in fact this mutation is in every cancer stem cell. And for the other mutations, we can also just compute in how many percentage of the cells. So now what we have this kind of knowledge about the coexistence of different mutation, and we know in how many percentage of cells they exist. So of course, for most of this data, we can build different trees resembling these percentages. This is just for example. Here, for example, for this data was 16 possible hierarchies that could be found. But fortunately, we have such data not only at the, so fortunately we have 2 time points. That's still very bad, but so we know also at the moment of the relapse. So we can now reconstruct trees from one data set, from another data set, and of course, since there is evolution between them, we can just neglect big part of them and then identify the real evolution. It is not always unique, but in fact, in most cases it is. So we developed such a combinatorial approach just to be able to transform the raw data to the information about such trees. Okay, so it means that for patients we could get this. On the other hand, we're thinking, can we learn more from clonal hierarchies? Does it tell me anything if I know if patient has this structure or this structure of the evolution of the cancer? And this, again, we approach with the model, but first in such a direct way that we simply wanted to see which structure are generated by our models under different assumption of the mutations of the parameters. So what we did our many compartmental model, and then we simulated this model with mutations. So it means mutation would mean that there will appear new clone with new A parameter, could be also A and P, but we can focus on A. And then we did just this distribution. We assume some distribution of this, how they appear, just computationally. And then we did statistics on results of our simulations. And what we wanted to understand, if depending on the structure of the tree, we have different, how the properties of the cells differ. Also, what decides how many clones we will observe at the moment of diagnosis, what it means, and also which, in fact, of these clones are the most aggressive. By aggressiveness, we defined if we would take such clone to a new environment, how well it would repopulate and establish the whole cancer mass. And again, so we did model, we're generating new ODEs, simulations, statistics on this. And it shows that while proliferation rates don't change significantly, the self-renewal depends on the position of the clone in the hierarchy. So it means if we have such a longitudinal trees, the cells downstream will have much higher self-renewal than this upstairs, and this will also be much higher than if we have short trees. However, the number of the clones at diagnosis does not depend linearly in a linear way on the self-renewal of the founder clone. So if the first mutation had very small self-renewal, then of course you will not see many clones, and in fact this will be not very efficient cancer dynamics. But also if the self-renewal of the founder clone will be very high, then you will not see many clones. And in fact, the story is that in this case, the cancer develops very fast, and they don't manage to develop more clones before the diagnosis. So it means that if patient comes and have few clones, one clone does not mean that the disease is not aggressive. It can exactly well mean rather mean that the disease is very aggressive, that he got high cancer mass with a small number of mutations. And then finally, we also looked at, in fact, if we have the same type of mutations, but we have different tree, that the time from the first mutation can be significantly different. So it seems that this how they mutate matters for the time dynamics. And finally, what we also would expect that, in fact, of course, downstream clones are more aggressive, so it means they have better repopulation properties. Okay, so this is just for understanding, and I just come to the comment on what we should now do with this. But before, I would like to say now to look at this from different perspective, because we said previously if we have model of this dynamics. So it is like, again, this equation is just without this integral. Then we have a selection. So what happens if we add mutation to this continuous model? And we can add mutation, of course, in a different way. It's still very phenomenological modeling. We can do it, as we saw on Monday in Camilo's talk, with diffusion. Here we use some integral operator to describe it with some kernels. Okay, so it can mimic diffusion. It can have also compact support. So as long as this operator has good properties, we can handle it. And we say we have here this scaling with the parameter epsilon. So we speak about small, or I would say rather rare mutations. And for this model, we kind of show that in fact mutations do not influence the selection process. So there is still selection to the clones with highest self-renewal. But of course, what we show here, but this is the same as in many approaches, is that we in fact show that we have convergence of the stationary problem to the measure located at this point. And additionally, for this model, we show that our stationary solutions are asymptotically stable. Just maybe very shortly that, okay, so for understanding this mutation, so here we just look at the stationary problem. And just you notice, okay, again, this is system, so a little bit complications. Again, I don't know. I don't know if one could bring it back to this approach with Hamilton-Jacobi equation. So at least due to this specific nonlinearities, I was not able to do it. But so here, what we do, we just formulate as a stationary problem, and if we can, from the first equation, we could find rho. So then it means the second equation is just equation depending on this parameter rho. And we can see it as a eigenvalue problem with eigenvalue lambda equal to 0 with such operator given by these two operators. And this can be reformulated to another eigenvalue problem with another operator with eigenvalue 1. So now if we show that there exists such rho, that lambda of rho is equal 1 is an eigenvalue of this problem with positive eigenfunction, then we have this existence of the stationary solutions. And this, so we do it with kind of infinitely dimensional version of Perron-Frobenius theorem. And so just, yes, so, and of course, so we look at the properties of this operator. Operator is compact, positive, irreducible. Then we can apply Rank-Rootman theorem, show this existence of this lambda and eigenfunction, and then we need to find also, so we know that this is now the spectral radius of this operator, and we know to find that there is such rho that we have this solution. And then again, looking at the properties of the rho, we can, and at the properties of this function R, we can do it. And then what we can show is the convergence. And again, we have this convergence to this Dirac mass in measures. And we have also convergence of this total mass, just, and we have, yes, so we have convergence for the stationary problem. And then what else we can show here? We can show stability of the stationary solution for the time-dependent problem. This is also a very technical thing, but this we can show. But it is, of course, still not showing directly that we have this kind of convergence in time to the Dirac problem, but numerical simulations, I did not put them here, but numerical simulations show indeed very fast convergence. Okay, so I'm nearly done. So first I would like to summarize this what we did. So in this projects, we always wanted to come as close as possible to this real data, real application, and to have ideas. Of course, it is still very far from real applications in the clinic. The clinicians love it, but of course, they would never come to idea to use it directly to the patients. But I think we are converging. So we showed that mathematical models, relatively simple models, provide possible explanation of the clonal selection, and moreover link the structure of clonal hierarchies to disease dynamics. And we can quantify properties that cannot be measured, even experimentally, not even dreaming about measurements in patients. These properties give us information about the course of disease. So it means, for example, when we could expect relapses. And also can predict patient survival. We have also other versions of models when we zoom out on some specific sub-processes like niche competition and compare them then with statistically with patient survival data, and usually it works pretty well. And just at the end, I thought, since we are at the beginning of the semester, and maybe I would like to tell you what are the open problems, not just technically, but conceptually, the problems which I find important. And it's not still that I don't have complete idea how to deal with it. One thing is, I said we can reconstruct this via combinatorics. I said we know that it matters for the disease dynamics. But how to use this information for patient? So now, additionally to this information on the blast counts, I would like to use this information about clonal structure, or evolution of clonal structure, to quantify the model. Okay, and to quantify this model or system of ODEs, of course, it's just secondary thing. So the question is how to use these structures, and in fact, whatever idea we have of using the structures in parameter estimation, you can think about some Bayesian approach with neural networks and so on, with all this efficient machinery, but still, what is missing? We need to quantify distances between different structures. And for this, we need a good concept how to deal with this. And the second, going a bit different direction, is that what's about solid? I mean, cancer in intertumor heterogeneity is one of the hot topics in oncology. So what about other cancers? So it shows that in solid tumors, heterogeneity is much higher. So you see a lot of clones. Of course, this picture is obtained using single cell data. So of course, with single cell data, you will see much more. We will see features even which are in small amounts of clones, or maybe not so significant. But the question is, can we understand this clonal heterogeneity in solid cancers? And in particular, what is interesting is that this heterogeneity changes between primary tumors and relapses, as in blood, but also somehow, if you have, for example, tumors which give metastasis to central neural system, for example, from lung, it seems that depending on the clonal structure of the primary tumor, we may hope to be able to say if there will be metastasis to the central neural system, or maybe other metastasis, or maybe no metastasis. So it seems that that could be information of very important for the prediction of the disease dynamics. But of course, these are now only first data and observation and ideas from the clinics, and we don't have models. For this, one would need to combine this clonal evolution models with solid tumors models. And if you think about our models of blood, in fact, the selection is due to this non-local coupling. So if you think that in solid tumor, there is much less communication between them because of the spatial structure, we may expect that there is more heterogeneity. And the question is if we could prove it. And okay, so here I just put collaborators of this main parts which I was talking today. So actually most of the work was done together with Thomas Still, who was PhD student and then postdoc in my group, and now is professor in Aachen in computational medicine. Also with another PhD student, Jan-Erik Busse. Also the analytical works with Piotr Gwiazda from Warsaw, with Tommaso, and with Silvia Quadrado from Barcelona. And also we work a lot with clinicians, and everything was possible due to the fact that Antoni Ho trusted us at the beginning. Thank you very much. I forgot. And just I wanted to say that the conference which we are organizing since 2 years will hopefully take place in 22. So, okay, the dates change, location change, many other things change, but it will be still joint event of European Society of Mathematical and Theoretical Biology and Society of Mathematical Biology in Heidelberg, and you should look at the web page, and we will, as you can imagine, many different problems. The world changed completely. Now it's for the many things in organization we had to start de novo, but we'll open the registration now soon. And yes, so everybody is invited to submit abstracts and sessions and everything. Thank you.
Thank you for the nice talk. I was wondering, with reference to the very first part of the talk where you mentioned that there is a hot bifurcation when you consider more than 2 compartments, what is the bifurcation parameter, or what are the bifurcation parameters there?
So you ask me now. So you have to change at least 2 parameters. I don't remember now. It was so far away from any reasonable parameterization that now I don't remember, but it was, you have to change at least 2 parameters. So, and of course, these are self-renewal. I mean, I'm not sure if you could do it only with proliferation rates, but it would be with self-renewal rates of the, or maybe self-renewal and proliferation. I can look it up, Thomas.
And the second quick question, if I can. In the last model that you showed us, the one in which you have phenotypic changes and a continuum structure, then there you have phenotypic changes only in the second compartment because you assume that only the cells which can proliferate can change their phenotypic state. Is it so? So you have
which changes?
The last model where you have the 2 equations.
Yes.
And the second one, you have an integral term that takes into account phenotypic changes.
Yes,
but you don't have it in the first equation.
Yes, because I mean, now I turn the order of equation, and this first equation are mature cells, so they don't divide, so they cannot mutate.
Okay. Yeah.
Okay, thank you very much. So, okay, maybe we can thank again Anna Marciniak and all the speakers of this morning session.